third-year biochemistry undergraduate student at the University of Liverpool. For my honours project with Professor Hasnain, I'm looking into protein interactions involved in motor neuron disease. In this video, I'll be talking about what motor neuron disease is, what experiments I've done, and what I've learnt. The disease is characterised by the loss of use of muscles due to degeneration of specialised cells called neurons that are responsible for muscle movement. I've recently just completed 12 weeks of research in the lab looking into two proteins known to be involved in copper transport that we believe may have effects on the development of motor neuron disease. Copper is taken into the cell via a specialised protein in the cell membrane called CTR1. This is then passed to a secondary protein inside the cell called CCS. These are the two proteins that I have studied interactions between. CCS then passes copper to a final protein called SOD1, which is involved in removing harmful molecules from the cell. SOD1 has already been proven to have effects in motor neuron disease. To look at the interaction between CCS and CTR1, I compared cells with normal levels of CTR1 to cells engineered to overexpress CTR1 in my experiments. The first major experiment I did was to show that CTR1 and CCS bind, done using an experimental technique called co-immunoprecipitation. Samples from both cell lines were taken and incubated with a bead that overexpressed CTR1 protein could bind to. We expected to see when analysing the data that CCS would be in the samples that CTR1 could be seen in, as CCS naturally binds to CTR1. Next, I looked into copper toxicity of the cell lines, expecting to see a difference between CTR1 overexpressing cells and normal cells. This was measured using colour change to represent the number of live and dead cells in the sample in response to treatment with different copper concentrations. With more time, this experiment would have been replicated 3 times to get solid and conclusive data. I have learnt a great number of skills in both practical experimentation and in how to interpret results. From my experiments, I can say that my cells were correctly engineered to overproduce CTR1, that from the co-immunoprecipitation it appears that CTR1 and CCS do bind naturally, and from the copper toxicity experiment it appears that copper toxicity correlates with the amount of CTR1 a cell is producing. Although these seem like little advances, I can say that I've learnt a great number of skills and have been driven to continue using my skills for further research. Thank you for watching.